So the last talk is on another set of genetic mutations that can cause autism, but actually which has been very challenging to study. These are inherited mutations that can cause autism, and specifically inherited mutations that are similar to mutations that cause cystic fibrosis or Tay-Sachs disease, where they can be inherited silently in families for generations, but which only cause disease if a child has the bad fortune to inherit a disabled copy of the same gene from both parents. And so these are recessive mutations. And our last speaker will be Klaus Schmitz Abe, who will talk a little bit about ways of finding recessive mutations by studying families where the parents are related to one another and therefore may carry the same sort of silent mutation. So Klaus is actually trained originally as an applied mathematician and has a PhD in applied math from Oxford University and has applied his mathematical talents to a variety of fields actually before moving to autism. He has used his math talents to study robotics, aircraft engines, and even modeling of financial markets. And recently, for the last 3 years, I think, he's been a postdoctoral fellow in the laboratory of Kyriakos Markianos at Boston Children's Hospital. And welcome, Klaus, and look forward to your talk.
Thank you, Chris, for this inspiring introduction. Good afternoon, everyone. It's an honor for me to be here in front of all of you, sharing my latest research to all of you, and especially for families and parents present here. I admire the families that are still here because it has been a long day. I don't blame you, you get sleep. Let's talk about a little bit about consanguinity, only to remark Chris say. Consanguinity comes from the Latin consanguinitas, that means blood relation. Consanguinities families are the families where the parents, dad and mom, are related in some way. They come from the same family or from the same common ancestor. If we plot the consanguineous families or the consanguineous marriage around the world, we can see in the red countries that are the highest consanguineous countries in the world. These families, there are 10 to 50% of the families are cousin marriage. And on the other hand, if we plot the same worldwide rate of birth defects and mental retardation, you can see there is a big similarity between these two plots. So, consanguinity and birth defects. Do you think this is a coincidence? Do you think this is random? No. My answer is no. When consanguinity plays, genetics play even a more important role because recessive genes, recessive defective births, have and play a big probability to appear in the population. So the second question I want to ask you for this audience is that, can consanguineous family help us to understand better autism? Okay, I think I will answer this question through my presentation. My 10 minutes remain. Our lab have created a pipeline that integrates all the databases and try to find the best genes, the best locus using probability and mathematical models. They try to explain a phenotype, in this time autism. Our pipeline, or autism pipeline now, integrates 10,000 samples. And the first data set I want to talk about is Simon Simplex, an amazing data set, and I have to congratulate the work they do. They phenotype more thousand, a lot of people all around the world. These families are 100% one affected family. What means that? That they require that the parents are unaffected, and they only have one child that is affected. Lately, they require that they have an unaffected sibling. The second data I want to talk is the AGREE data. And again, this is an amazing consortium that they did an amazing job. The difference about AGREE and the Simon simplex is that the AGREE data is multi-affected family. That means that 88% of the families have two or more affected. The third and last autism data set is the WASH lab data set. The difference between the other two is that this data set is consanguineous. I think an important property to look for recessive genes. We have 200 families and 242 samples. Even if the sample is small, I will show you that it's unique. As a control, everyone knows HubMap. HubMap is a great data set that was collected like unaffected simply. It was collected to normal people. So everyone uses it like a control. We have 1,200 samples for 11 populations and 4 continents. If we calculate the ratio, the male-female ratio, we see that the Simon simplex is 6.4, AGREE is 3.7, and the WASH lab is 4.6. Why we have a different female ratio? Because they are different. Simon simplex is one affected, AGREE is multi-affected, and the WASH lab is consanguineous. They have a completely different genetic architecture. To prove that, if I divide it in one affected and multi-affected, and consanguineous and not consanguineous, we can see now that the rates start to coincide. I have to add that multi-affected and consanguineous families behave similar. I don't believe they are the same. Conclusion of the slide. One affected and multi-affected families, consanguineous and not consanguineous families have different genetic architecture, behave different, and have different patterns with respect to CNV. Before I start to talk about de novo, I would like to do clear definitions for you. Let's start. Mom and dad met, they get in love, and then they get married and have a baby. If we go to their genomes, we know that mom have 23 pairs of chromosomes, and dad have 22 pairs plus X and Y. Let's do an example. Let's take only chromosome 1 that have more than 2,000 genes. So mom have a pair, one comes from the mom, and the other one from her mother, for her father. And the same story for the dad. If we go to the genome of the baby, we see the same story. We have one chromosome that comes from the combination of these two, and the other one from the combination of the dad. This combination is unique. And that's the reason you are different to your siblings. If we look carefully, this is not exactly what happened. There are little pieces that they don't come from father or the mother. They are deletions, they are insertions, they come from something else. We call that de novo events because we don't know where they come from. De novo events are random, and because we can find them in all chromosomes. We can find them in affected and unaffected, and they are very important for evolution in the human being. Some babies have small amount of de novo, and some babies have a lot of de novo. this excess of de novo have been correlated with age of the parents, mainly with male, place of living, food and stress, and smoking and drinking. This is the bad part. If that deletion, if that de novo event kill an important autism gene, the baby have a high risk to become autism. And that is what we call de novo autism. This is not new. Autismo and de novo have been correlated in the old times. There are publications since 1982 where they start to say there is a lot of de novo in autismos samples, or in autismos kids. Although, until 2010, Pinto, using 2,100 trios, just European, they claim and they did the first strong statistical evidence that there is an excess of de novo in autism child. After that publication, Sanders and Levy followed this research using the Simon simplex, and they claim the same. They use different technologies, that's the reason the rate is different. Now that we are in elections, we can do a pool of pools. So, just to say, it's just to show you that there is at least 3 times an excess of de novo in an autistic child compared to normal people. Let's do some maths, but I am here. Let's use the Simon simplex that there are 1,000 affected, and let's plot the de novo average per sample, where the red bar are the affected or the autism children, and the blue bar are the unaffected. We can see that there is no big difference. There is no difference between these two. Why? Because de novo are common. We can find them in affected and unaffected. And even if we go to the control data that we know is HapMap, that even if they are using a different technology, we see that we have 2.92 de novos per sample. To replicate what Levy and Pinto did, we need to find rare de novos. What is rare? Rare de novos are CNVs that are not common in the population. So if we subtract the common, all the CNVs that HapMap have to our database, the story comes different. Now we see that there is a difference between the red and the blue bar, and with a p-value of 0.031. And then if we go to de novo bigger than 100 kb, we get even a better value. On the other hand, what happens if we do exactly the same analysis with consanguineous families? We see a trend. Definitely we see that there is a difference between the affected, but we don't see any p-value. Maybe this p-value, we don't see the p-value because we have a small amount of samples. However, we know that this data set are consanguineous and some are not consanguineous. So if we split both data sets, the story becomes clearly now. We can see that not consanguineous people replicate completely what the Simon simplex has. And the consanguineous people doesn't show any different. So there is, for consanguineous people, we can say there is no mainly driving by de novo. Conclusions. De novo events plays an important role for one affected non-consanguineous families. After I finish with de novo, I will start a new story. This is a new story. This is copy number 0, homozygous deletion. these results have never been implicated or published before. Let's do only just clear and define. We have mom, dad, again the baby. This time the mom have a pair, but this time the mom, one of the chromosomes have a deletion. Again, the dad have the same story. If these deletions only delete recessive genes, that means that you only need to have one copy to do well, mom and dad will be normal. What happened with the baby? If the baby is unlucky and inherits these two deletions, that we call, in our say, we call homozygous deletion, because they delete the same locus in both homologous chromosomes. And this deletion delete, again, genes that are important in autism. The baby have a high risk to become autism. And that's what we call inherited autism. Doing exactly the same analysis that I did with de novo events. And now, instead, I plot de novo, I plot homozygous deletion. Only just this deletion, copy number 0. We see for the Simon simplex and for the agreed data that there is no difference. There is no p-value. And this is what we expected. They are not consanguineous. If we plot the wash lab that are consanguineous, we see a really clear signal, a big difference between the affected and the unaffected. And this is a p-value of 0.037, and the other p-value is 0.14. And again, if we subdivide it, again in consanguineous and non-consanguineous, again, non-consanguineous replicate, I expected, what the Agree and the Simon simplex did. Replication is very important in mathematics and in genetics and in many areas. And then, conclusion. Consanguineous, for this, copy number 0 or homozygous deletions play a very important role for recessive autism. We couldn't find these results without consanguineous family, sort of the WASH data. And again, it validates Chris' argument that the relation between consanguinity and recessive genes are very important. This is my last slide. I promise. But let me conclude to you. Using the approach that we have with the homozygous deletion copy number 0, we have selected and we have now 23 strong candidates for recessive autism. I want to mention, everyone asked me to mention one gene. One of the genes I want to mention is PSD3. I like the name of the gene. We found a homozygous deletion in the 3 data set, in the WASH lab, in 2 affected, in a family of 2 affected in the Agree, and 1 family in the Simon simplex. Because it replicates in the 3 data set makes us feel that it's a very more strong candidate. And another point is just recently in the Neuron Press, they described this gene like is a transcription factor high expression in the brain in the postsynaptic density and required for synaptic plasticity. So it's clearly a brain gene. Well, after we have selected all the genes, we go to test the genes in function analysis and animal models. Oh, this takes years, 3 to 5 years to do that. All this research, all this year, is for one reason, is for one goal, is to understand autism better, to understand the biology and genetics behind that, for only one reason. So for in the future, doctors and hospitals can improve the quality of autism. And why not? To find a treatment for autism. I didn't come here to lie you to say that we have the answer. I didn't have to say that we understand perfect autism. But I guarantee to you that many people around the world, many people are not going to stop thinking about the problem until we came to you with the right answer. You deserve it. Thank you.